Hello everyone. In this video we are going to discuss the control of the cell cycle. In previous videos we explained about cell cycle, the stages of cell cycle, the phases of mitosis, the interphase, the differences between chromatin, chromosomes and chromatids. Now a very important question we ask, how cell cycle is controlled. But before we ask this question, let's discuss why it is controlled. So cells divide, speaking about human body, our cells divide based on the body needs and based on certain signals. For example, at the different stages of growth, cells can activate or inactivate their division in order to meet the needs of the body. So before we start discussing what are the factors that control cell cycle, let's have a recall about the stages of cell cycle. So first of all, we know that the cell cycle is divided into 3 stages. Let's recall these stages because we will need them in order to understand more the control of the cell cycle. So in this pie chart, I'm going to write the phases, starting with G1 phase. Then we have the S phase, the phase where DNA is replicated. Then G2 gap 2 phase. Then we move to mitosis. I'm not going to mention all the phases. We have prophase, metaphase, anaphase, telophase, and cytokinesis, the last stage in the cell cycle. Okay, if I assume that the cell cycle is taking 24 hours in human body, so I might give 8 hours for G1 and 7 hours for S, 6 hours for G2, 2 hours only for mitosis, and 1 hour for cytokinesis. So I will keep this aside. Because when we speak about control of the cell cycle, it will happen at different phases, at different levels of the cell cycle. So let's start speaking about the control of the cell cycle. First of all, the cell cycle is controlled based on 2 different factors: the internal and the external factors. Let's start with the external factors that control cell cycle. And here, internal factors. Okay, the external factors that control the cell cycle are divided into 2: physical and chemical. Physical factors and chemical factors. Now, before we continue, it's important to know that when I say control of cell cycle, I'm speaking about either activation or inhibition, either activating cell cycle or inhibiting it, stopping it. So whether the internal or the external factors will be working as a switch in a circuit, either turning on or turning off the cell cycle based on the body needs and based on whether the cell is ready to divide or not ready. So let's start first of all speaking about the physical factors that control the cell cycle. Now, when I say physical factors in the cell, mainly I'm speaking about the physical area, the place where the cell is located. And here we can explain this part by looking at cells dividing. It's important to know that cells divide based on the body needs, and one reason for cell division might be repairing tissues. So when cells repair tissue, they have to divide in order to create new cells. So physical factors are related to the area, to the space where the cells divide. And this is controlled by proteins located at the plasma membrane or the cell membrane of the cells that allow the cell to adhere or to connect with the cells around it. So every cell, in this example here, every cell, if I look at this cell for example, it is connected to 1, 2, 3, 4 cells around it. And this one is connected to 4 cells. Physical factors will either allow the cells to divide if there is enough space for them to divide in a plane or one layer. If there is not enough space, the cell will halt, or cell cycle will halt or will stop. As a result, cell cycle will stop. So physical factor, we can call it cell to cell contact. Cell to cell contact. If the cell is connected to cells around it in the same layer, division will stop. Okay, as a result, and they have done an experiment. They put actively dividing cells in a petri dish in the lab under certain and optimum medium. And these cells started to divide and divide and divide, and they were looking at how many cells are formed. After a certain amount of time, cells stopped dividing when all the petri dish was filled of one layer of cells. Division stopped. And this is called physical control on cell cycle. Now, what will happen if this physical control is not working? And this is the case of uncontrolled cell growth. This is abnormal case, usually happens when there is cancerous cells. Cells won't respond to the physical factor. Even if they are surrounded by cells around them, they will keep on dividing. As a result, there won't be enough space, so they will start growing more layers and more layers. And this is what we call a tumor. If we are talking about, for example, cancer, this is called a tumor. So the absence of physical control on the cell cycle might lead to formation of tumor, and this is the case of uncontrolled cell growth or cancer. So this is first the physical control on the cell cycle. Now let's move to the chemical control, and still we are talking about external factors. Chemical control, it has to do with signals, external signals that are received by the cell. So if I consider this is the cell, okay, DNA, whatever. At the surface of the cell, there are membrane proteins. These membrane proteins, some of them act as channels, some of them act as pumps, and some of them act as receptors for external signals. Mainly these external signals are usually hormones, okay, or proteins, or other proteins. By the way, hormones are proteins, but not all proteins are hormones. So these hormones, we call them growth hormones, and their main function is to bind to the cell, and as a result of this binding, they will initiate other signaling inside the cell that will activate cell division. So growth hormone, as if there is a messenger that comes to the door and delivers a message and leaves. This message is activating the cell cycle or even inhibiting it. An example of growth hormone are all the hormones that initiate the release of sex hormones during puberty. And this will lead to more changes in the physiological structure of the body. So chemical signal, mainly we speak about GH or growth hormones that bind to the cell surface. As a result of this binding, they will cause more activation to other molecules inside the cell. So we have internal or intracellular signaling and extracellular signaling. Extracellular signaling is a signal that is received from outside the cell. So to understand it more, let's consider this is a cell and this is a receptor here, and this is a hormone, growth hormone. And I will add one more molecule here. Okay. So the binding of this hormone to this molecule will lead to the activation of this small internal signal that will move in turn and activate cell division. This is an example how hormone or growth hormones can activate cell division. So this is an external signal. It doesn't go inside the cell. It will only bind to a receptor protein. This is the receptor, and this is the growth hormone, and this is here internal target molecule or target protein. This will move inside after it is activated, and it's going to activate cell division. So in brief, the external control of cell cycle, either physical or chemical, can either activate or inactivate cell cycle. Example on physical control is cell-to-cell contact. One example might be when you have an injury in the skin and you lose some cells of the skin. So your body, the epithelial cells in the skin, will initiate cell division, but this division will stop after all the cells cover the injury, cover the cells that were lost. An example of chemical signal, it happens at puberty when hormones, growth hormones, are released and they activate cells that are related to the reproductive system or the release of sexual hormones that will have changes in the body. For example, in males, growth of beard and maybe bone elongation, etc. These two factors, they have to do nothing with what's going inside the cell. They only act based on the cell surface and surrounding of the cell. Now, moving to the most important control, which is the control of cell cycle from inside the cell. So, in order to understand more the control of the cell cycle from inside the cell, we have to go back to the diagram of the cell cycle. Control of the cell cycle happens at different levels. The first level, it happens at G1 here. So here we have the first control. Okay, here we have the first control we call it G1 checkpoint. The second checkpoint happens at G2, we call it G2 checkpoint. And the third checkpoint, it happens during M, we call it M checkpoint. So we have 3 checkpoints that control the cell cycle, and we consider them as internal factors that control cell division. Okay, what happens during these 3 checkpoints, and what are the factors, the proteins that have a function in this control? This is what we are going to explain now. But before we start, let's revise what happens during G1, S, and G2 to understand what is the function of every checkpoint. So speaking about the first checkpoint, G1, the G1 checkpoint, it has to check what happens during G1. And mainly, this checkpoint is going to check for cell size. It's going to check for any damage in the DNA. It's going to check for organelles. Are the organelles copied? Everything going right? If all of these are checked, then the cell will proceed to S phase. During S phase, DNA is replicated. And after DNA is replicated, the cell moves into G2, where it undergoes further growth. And here we have the most important checkpoint, which is G2 checkpoint. G2 checkpoint will check for any mistakes in DNA replication. This is a very important step because at this level, if there is any mistake in the copying of DNA, as a result, we will have 2 daughter cells that have damaged DNA or different or mutations in the DNA. As a result, they won't be normal cells. So in addition, it will check for the cell size. And it will check if the cell is ready to start mitosis. But this is the most important event that happens during G2 checkpoint. Now, if this is checked, then the cell will proceed into mitosis, and the M checkpoint, it's going to check mainly for the binding of spindle fibers, the proper binding of spindle fibers to chromosomes. As you know, during mitosis, spindle fibers align the chromosomes in the middle of the cell, then they are separated. So this checkpoint is very important to ensure that in humans, 46 chromatids move to one side and other 46 chromatids move to the other. If there is any mistake, sorry I had to stop, but the birds were very noisy, so I had to take them to the other room. So yeah, I was speaking about the M checkpoint. This checkpoint will check for the proper binding of the spindle fibers on chromosomes or to chromosomes. This is very important step because if chromosomes are not separated evenly, if one cell cannot get 46 and the other 46, as a result, this will lead to cells that don't have the same number of chromosomes or same amount of information. Now, this might be very critical if this mistake in the separation of chromosomes happen in meiosis or in the production of sperm and egg cells. As a result, the embryo might have trisomy or monosomy, either missing one chromosome or having extra chromosome. One example of trisomy is trisomy 21 or Down syndrome, where chromosome 21 has 3 copies instead of 2 copies. And another example on monosomy is Turner syndrome. In Turner syndrome, the embryo will have 45 chromosomes instead of 46. All these abnormalities might exist if there are mistakes mistakes during the mitosis, mainly during metaphase and anaphase. That's why we have the checkpoint that will always make sure that the separation of chromosomes is happening evenly and there are no mistakes. In case there are mistakes, then the cell will stop dividing, or simply it will undergo apoptosis or self-destruction. Now, this is in general the function of every checkpoint. But what are the key factors that act in the internal control of the cell cycle? This is what we are going to discuss now. So let's take, internal factors. So again here, the internal factors will either activate or inhibit the cell division. We will start with activation of cell division. I mean by activation, if everything is going well, there is no damage in the DNA were copied well, the cell size is correct. What are the factors that allow the cell to proceed from G1 to S to G2 to M? Let's start with activation. Okay, to start with activation, let's ask what are the molecules that activate cell cycle, or what are the molecules that act during M, G1 and G2 checkpoints. Actually, there are two classes of proteins. We call them cyclins and cyclin dependent kinases. So cyclins, and they are of 4 types, and CDKs, or mean cyclin dependent kinases. So these are two different proteins that work together in order to activate cell division. And how they work together. In order to understand this type of control, we have to start by the first step of this control. The first step is, in order for cell cycle to be activated, cyclin must bind to CDKs, cyclin dependent kinases. This is number 1. This is the first step. When they bind together, they will form a complex. So let's assume that the cyclin, the green color, and the CDK with the red color. In order for the cell cycle to be activated and move from one checkpoint to another, the first step in this activation is the binding between the cyclin and CDK to form a complex. Now, but this is not enough. Inside the cell, there are enzymes that are called kinases. Those are enzymes that activate target molecules. Kinases are like enzymes that have keys, and without these keys, many reactions cannot happen in the body. So the function of kinases, it's a huge family of enzymes. Their main function is to give this key for any reaction or any step to happen in the cell. What is this key? This key is called phosphate group. And chemistry students can understand what is a phosphate group. A phosphate group is a phosphate which is linked to oxygen molecules that will form a functional group that can influence molecules and activate them. Now, as biology, you don't have to understand the structure of phosphate, or what you have to know that this complex, it has to bind to a phosphate group to form a new complex. So let me take a copy of this. Sorry. 1 minute. Yes. Plus a phosphate group. So this is the second step. So we have the cyclin CDK complex. Then we have an enzyme called kinase, which is donating a phosphate group to this complex. As a result, now we are having 3 parts or components, which is 1 phosphate, 1 cyclin, 1 CDK, cyclin dependent kinase together. What is the last step? The last step in this control mechanism is this complex will lose or will donate its phosphate to a protein. We call it the target protein. Now, at this level, at the school level, we don't have to know what is the name of the target protein and how it works. But all what we have to know that this target protein, it's the one that will activate cell cycle. And for this target protein to be activated, it needs to be phosphorylated. It means phosphorylate, it means it needs to bind with a phosphate group. And this is the function of the complex. This complex will add the phosphate group to it. As a result, it will be activated. So without the phosphate group, without the P, this one cannot be activated. Now it is activated. So let me go back. This is the third stage. So the phosphate group is coming from the cyclin CDK complex that received a phosphate from kinase. Now, some of you might say, okay, why this is very complicated? Why the control of cell cycle? And it's not only like that. There are other reactions in the body. We call them metabolic pathway, like many reactions happen, step 1, step 2, step 3. Why it's not simply one step and that's it? This is very important because if this reaction happens only by one step, there is high chance that controlling it might be very difficult. Having different steps, controlling these steps can happen at different levels. For example, if there is no kinase, this will stop. If kinase is there but cyclin is blocked, this process will stop. If cyclin is there and kinase is there but CDK is not there, again this process will stop. So in order to control cell cycle, to make sure that there are different ways of controlling it, these steps are important. And we can see an image about these steps that might explain it in a more clear way. Okay, let's look here. So this is the cyclin. This is the cyclin, and this is the CDK, cyclin-dependent kinase. The first step is they bind to each other. The second step is they receive phosphate from an enzyme called kinase in order to form cyclin-CDK complex, which is phosphorylated. Phosphorylated, it means a phosphatic group is added to it. And one more time, phosphatic group, it's a functional group. It means it can make a function in the molecules. Let's continue. And the last step is this cyclin-dependent cyclin phosphate complex is going to bind to a target protein and donate or give this phosphatic group to the target protein. Now this target protein is activated and it is going to do activation for the cell cycle. I hope now it's very clear. So all of this is needed in order to give this phosphate to the target protein. The target protein, it is phosphorylated now. It can do activation for the cell cycle. There are different types of cyclins, and these types of cyclins, they work at different phases, but all of them work in a very similar concept. They have to bind to cyclin dependent, and they have to activate a target molecule. So four identified types of cyclins. There is cyclin D, E, A, and B, and they work at different levels. Like for example, cyclin D, it is activated during G1, S, and G2, and even in mitosis. Cyclin E mainly, we can relate this. It is activated in G1 checkpoint. In G1, cyclin A is activated in G2. Cyclin B is activated in M, and we have cyclin D. It is activated in all checkpoints. Okay, these are the types of cyclins, and this control here, it is the activation or the positive control of the cell cycle. Now let's move to the second type of control, which is inhibition of the cell cycle. So simply, inhibiting cell cycle, it happens at two levels. The first level, either it happens by inhibiting the CDK, and we call them CDK inhibitors. So we have CDK inhibitors that prevent the binding between CDK and here it will prevent the binding between these two. As a result, all the rest of the steps won't happen. And the other molecules are called inhibitory regulators. And the other types of proteins are called negative regulatory proteins. These are the two classes or types of molecules that inhibit the cell cycle. We are going to explain briefly about each one. Let's start with CDK inhibitors. CDK inhibitors, simply, they prevent the binding, as we mentioned, between cyclin and cyclin independent kinases. Let's see how does this work. So if CDK is inactive, then no phosphatic group is going to bind to the target protein. As a result, the S phase is off, so the cell won't proceed to G2 and M. This is when the cyclin is there, and this part mainly is related to the activation, as can you see here. If cyclin binds to CDK, they will be able to give phosphate to the target proteins that will activate the S phase and the rest phases of the cell. Now, how these regulatory proteins work, negative regulatory proteins work. P53 is an example of a negative regulatory protein. We call it P53. And it has another name, tumor suppressor protein. Suppressor, it means inhibitor. Tumor suppressor, stopping the tumor. How this works? P53 will activate the CDK inhibitors. As a result, the CDK inhibitor will prevent the binding between CDK and cyclin. As a result, the complex isn't activated and there is no inhibition. So the negative regulatory proteins will activate the CDK inhibitors. So the point is simply, in order to control cell cycle, in order to prevent cell cycle from happening, we need to prevent the binding between cyclin and CDK. In order to prevent that, we have a molecule called CDK inhibitor. This molecule must be activated. In order for this molecule to be activated, we need other proteins called negative regulatory proteins. And there are three types of these proteins. There is P53, and there is another molecule called RB, which means retinoblastoma proteins. And there is one more molecule called P21. So the negative regulatory proteins are divided into three proteins: P21, P53, and RB proteins. All of them are called tumor suppressor proteins. And all of them, they inhibit the binding between cyclin and CDK, as a result, stopping the cell cycle. What is the importance of this? Is stopping the cell cycle something bad? No, definitely it's not. Why? Because the absence of inhibition will lead to uncontrolled cell growth, will lead to cancer, will lead to formation of tumors. So the tumor suppressor proteins, and again, the name implies its function, tumor suppressor. The proteins that suppress, it means to stop the formation of a tumor. So if there are mistakes in the genes or mutations in the genes that form these proteins, as a result, these proteins won't be activated. As a result, the cell will be active all the time. And this will lead to cancers and other problems in the body. So the question is, when do these are activated and how can they control cell cycle? Let's go back to these stages here. So we said that during G1 there is a check for DNA damage. What if during G1 the DNA was damaged? So during G1 checkpoint, DNA was damaged. What will happen? If there is a damage in the DNA, the enzyme or the protein P53 is going to be activated. And activation of P53 will lead to two choices. Will lead to two choices. The first choice is either giving the chance for the cell to repair the damage, and restart the cell cycle. This is the first option. And again, here there are more proteins that are involved in the process of repairing, including enzymes called DNA polymerases. These enzymes have the ability to repair mistakes. And the other choice is, right here, or if this is not possible, P53 can activate something called apoptosis. What is apoptosis? Apoptosis is programmed cell death. Apoptosis is programmed cell death, like the cell is committing suicide. So if the damage in the DNA is not repaired, then the P53 will allow the cell to destroy itself so that it won't proceed in cell division. And both of these steps are very important to prevent the production of abnormal cells. This is one example of how the negative regulatory proteins such as P53 can help or can save the body from having uncontrolled cells, abnormal cells, and maybe these cells can become cancer cells. So to recap all what we have explained quickly, let's go back to our title, control of cell cycle. So cell cycle can be controlled by external or by internal factors. The external factors include physical and chemical signals. Physical has to do with the cell-to-cell contact. If there is not enough area to divide, the cells will stop dividing unless if there is something abnormal going on. And here when we speak about tumors. Chemical external control, it happens when hormones called growth hormones bind to the cell surface and activate cell division. Internal factors that control cell division are divided into two sets of proteins called cyclins and CDK, and those activate cell division. And the other sets are called CDK inhibitors and negative regulatory proteins, and those together they inhibit cell division. For the cell to be controlled, both are needed. In terms of activation, cyclins must bind to CDKs, cyclin-dependent kinases, in order to form a complex. After that, this complex must receive a phosphate group from an enzyme called kinase, and after that, this phosphate will move or will be given to a target protein. The target protein is going to activate cell cycle at different phases of the cell cycle, including G1, Gs, and G2 and down. Without the presence of the phosphate, the target protein won't be able to be activated. And there are 4 types of cyclins. Every type of cyclin work at a specific phase, and there is one type that is activated during the cell cycle, which is cyclin D. Moving to the inhibition, CDK inhibitors prevent the formation of the complex between cyclin and CDK, and CDK inhibitors are activated by negative regulatory proteins. There are three types of negative regulatory proteins in humans: P21, RB, and P53. RB means retinoblastoma proteins. This protein, in addition to P53 and P21, they are also called tumor suppressor proteins that prevent the formation of tumors. They stop uncontrolled cell division. So they have a very positive and important effect in the body. How they do that? Simply by activating CDK inhibitors. CDK inhibitors, they prevent the formation of the complex. As a result, stop the cell from dividing. One example is when the DNA is damaged during G1, P53 will be activated, and P53 will try to repair or will try to activate the repair mechanism. If the damage of the DNA cannot be repaired by other proteins, then P53 will activate apoptosis, which is programmed cell death or self-destruction of the cell. As a result, there won't be abnormal cells to be produced. Thank you so much, and if you do have any question, please put the questions in the comments, or you can send me an email. I will drop my email address in the comments below. Have a good day.